everyone. In this video, we're going to be talking about tissue injury and repair. And I think it's a really great time for this video because it's going to enable us to synthesize some of the concepts that we've been working on over the past several videos. Everything from mixing different tissue types so that we can see structures and organs and how they come together, to what types of cells we might find in the various tissues, as well as even back to talking about different types of receptors and the concept of having a receptor, a protein type of receptor on a membrane, and like what the protein function is, and even a little bit of membrane transport. So I think it'll be a good review, but also a good application. So let's start with a little story. And let's say that it is a beautiful afternoon, 80 degrees, nice and sunny, and we're walking outside in the grass barefoot when all of a sudden, oops, we step on a rusty nail. That hurts so bad. And also there's a lot of other things that are going to happen, right, besides the pain. And that includes bleeding and swelling and maybe a risk of infection and lots of things that we probably kind of already know. But in this particular case, what we really want to do is take a look at what is happening on a cellular level and really see inside of the body. So we're going to say that the bottom here is the skin, and that is the bottom of the foot. So we're just kind of looking from the bottom of the foot up at everything that is going on. First, we have our epithelial tissue, we have our connective tissue, sort of a similar model to how we drew it in the last video on membranes. And then we're going to draw the blood supply. So we know that just below the surface of the skin, we have some blood vessels. We talked about filtration. We talked a little bit about arteries lead to arterioles, which lead to capillaries, which lead to venules, which lead to veins, which bring the blood back to the heart. And in this case, we'll be able to label those structures, arteriole, capillary, and the venule. And the venule is a post-capillary venule. And that's important because some of the stuff that's going to go on as far as how the body is responding to this injury are going to take place in the post-capillary venule. So we really want to pay attention to that. Now, initially, we drew the structure just kind of with some basic lines, but I really want to focus a little bit more on the actual structures that we see. And we mentioned recently in our cell junctions video that we have endothelial cells, which are simple squamous epithelial cells that are lining all of our blood vessels. And in the case of a capillary, that's all we have. There's not any more depth really other than those. And the same thing is true in the post-capillary venule. We have pretty much, with a few exceptions, just a single layer of these endothelial cells. And that's really where the important function lies. So we're just going to kind of take a look at all of them. All right, so now let's say that the nail has penetrated the epithelium and the connective tissue, and because it's in the connective tissue and we happen to have a blood supply there, we also puncture the capillaries. So of course that's going to lead to bleeding. That makes a lot of sense. And I think it also would make sense to say that depending on the depth of the injury, we would see different types of damage. So in this case, we're deep enough to get some blood release from the capillaries. If we were deeper, we might see other tissue structures that we're not mentioning right now. And if we were less deep, like sometimes just a simple paper cut might cut through the epithelium and not really into the connective tissue, and therefore we might not get any bleeding, right? Maybe not. So the depth of the wound is definitely going to affect how badly we're damaged. All right, so now that we have our injury, the first thing that's going to happen in response to the injury is inflammation. And inflammation is characterized by 4 general things. Redness, which is the medical way of saying that would be erythema. So those 2 words mean the same thing. Warmth, swelling, and pain. And so what we're going to do is talk a little bit about how each one of those things occurs. The first thing that we're going to look at is the mast cell. And we know that we have mast cells in the connective tissue, right? We talked about those before. And the mast cells, we said, contain granules of histamine and heparin. And in particular, what we're going to focus on right now is the histamine. One thing that's going to happen in response to this nail puncture is the mast cells are going to release histamine. And the mechanism for releasing the histamine is exocytosis. Now, it just so happens that we have a receptor for histamine on our endothelial cells. So we have lots of histamine receptors in our body, and so we just abbreviate for histamine, we use the letter H, and we have H1 receptors and H2 receptors and H3 receptors and H4 receptors. So there's a bunch of different kinds, and usually it will be a certain kind is found in a certain place. And it just so happens that the H1 receptor is found on the endothelial cells, and we're really targeting the post-capillary venule, as we mentioned. So the histamine, after exocytosis, is going to float on over, and it's going to bind to the histamine receptor. And in this case, what's going to happen, we're going to kind of skip the actual stuff that's going on inside the cell that enables this to happen. We're just going to cut to the chase and say, here's the outcome. The outcome is that we cause contraction of the endothelial cells. We cause them to kind of shrink up a little bit and pull away from each other. And because they pull away from each other, it means that now there are gaps. There are gaps between the cells that we didn't have or didn't really have before. And it's true that, even in capillaries, we have the ability of some stuff to squeeze between the cells as long as we don't have tight junctions, right? We talked about that. That would be paracellular transport between the cells. But in this case, because we bound the histamine to the receptor, we are causing a more dramatic effect than we would otherwise normally have. And so now we purposefully can see that we have a nice big gap for some stuff to leak out of the blood vessel. So what we say is that histamine causes increased vascular permeability. So permeability refers to the ability of stuff to move either across a membrane or from one compartment to another. And in this case, what we're talking about is the ability of the fluid, so the plasma inside of the blood vessels, to be able to pass out of the blood vessels and into the connective tissue that surrounds it. And so when we increase vascular permeability, we're making it easier for that fluid to leak out into the connective tissue space. And so what we're going to get is swelling. So that's one of our 4 key parts of inflammation that we mentioned. All right, so let's talk about a few other things. So something else that happens in response to the histamine is we have vasodilation. And vasodilation is the widening of the blood vessels, and that causes the blood vessels to essentially engorge with blood. So there's more blood flowing to this area. And there's lots of reasons why we would want more blood to flow to the area, including making sure that we're able to bring various types of white blood cells, which are protective, to the area so that they can gobble up any kind of bugs or bacteria or anything that's not supposed to be there. And we'll talk more about that in a second. But as a result also of vasodilation in the skin, that's going to bring the blood vessels actually closer to the surface of the skin. And so the vasodilation accounts for both the warmth and the redness. We're bringing the blood vessels, filling them up with blood and bringing them closer to the skin. And so that brings us to 2 other of our key points of inflammation. And then the last thing that we want to mention as far as inflammation is pain. And we know that inflammation is very painful, and there's a couple of different reasons why that is. One of the reasons is because of the synthesis of something called prostaglandins. And prostaglandins are a specific type of chemical, and one of the things that they do is they signal that there's pain. And the thing that maybe you already know, or that you could most easily connect to with prostaglandins, is when you take let's say, in this case we have this foot injury, right? And I don't know if Advil or Tylenol is really going to cover this nail in the bottom of this person's foot. But let's just say that, we were going to, and if you're taking Tylenol or Advil or something, and, the question is, well, how does it know where the pain actually is in the body? How does it know how to get there? And the answer is because of these prostaglandins. And so we actually, when we take those medications, we're actually kind of preventing the synthesis of prostaglandins. So that is what leads to the reduction in sense of pain. But the prostaglandins, so it's a little bit of an oversimplification, but the prostaglandins are helping us to sense this pain. And the other thing is, and we know that we have neurons, and we haven't talked about neurons really in much detail, except to say that they exist, and they certainly will exist in this connective tissue space. We have nerves, we have neurons. So sensory neurons are neurons that sense various things, including pain, and so that's another way that we can communicate to our brains that we're experiencing severe pain as a result of this nail. All right, now the last thing that I want to talk more about is basically the types of white blood cells and other cells that are handling the fact that because we have this nail that has pushed into the skin. One thing that we would immediately probably be worried about with a rusty nail is tetanus, right? But any kind of bacteria or virus or any kind of pathogen, something that's bad for the body, is something that we would be worried about and that the body wants to protect against. And there's a couple of ways that we do that. So macrophages are a type of cell that are often hanging out already in the connective tissue space. So this macrophage is already here. And if there are any pathogens, like these little green guys, if there are any bacteria or anything that we don't want, they're basically going to phagocytose. So just kind of like wrap their parts around and engulf this pathogen, and then they'll be able to chew it up and digest it, and it won't be able to cause harm in the body. And what we really are interested in is preventing those pathogens from squeaking into the blood vessels so that they can then circulate and then proliferate or grow and make more of them and cause a larger problem. We really want to keep this as a local, quickly managed situation. So macrophages are there and they can gobble up those pathogens. There's a second type of cell that I don't think we've ever mentioned before, and that's the dendritic cell. And the only reason I'm mentioning it now is because later on when we talk about the immune system, we're going to kind of revisit this whole topic of tissue injury and repair, and we'll be able to fill in a few other things that are happening in more detail, especially regarding how we manage those pathogens that are potentially invading. And so the dendritic cell is also able to recognize these pathogens. It's also able to engulf them, and it's also able to release special chemicals called cytokines. And those are chemicals that are able to basically direct other cells to do specific things. And I'm going to give you one example. And the reason also for mentioning this is because down the road, we'll talk about different types of body responses that involve cytokines, and also various drugs that we might administer to patients when there's a particular issue, like too many pro-inflammatory cytokines, so too much inflammation or too much something, we can try and fix it. All right, so one thing that these cytokines are able to do is attract more white blood cells. So we mentioned that one thing that can happen in connective tissue is we can have cells that maybe aren't in the connective tissue, but we want to bring them into the connective tissue when there's a specific problem. And in this case, we have a nail under the skin, so that's a big problem. So therefore, we're going to attract some more white blood cells. And the example that I'm going to use is neutrophils. So in that same connective tissue video, we mentioned the different types of white blood cells, and neutrophils are pretty much the fastest to arrive when we have any kind of event like this. So the neutrophils are cool because they actually just roll along the endothelial cells. And as they roll along the endothelial cells, it's kind of described as if it's a tennis ball rolling on Velcro. And then when there are these cytokines, or when there is this whole big mess that's happening that says, oh my goodness, we need to have some more white blood cells to gobble up these pathogens before it gets into the bloodstream, well, in that case, the endothelial cells are actually going to pop out these receptors for those neutrophils, and it will grab onto them. And so what we have actually is an interaction between a carbohydrate that is on the neutrophil, which we said carbohydrates sometimes in the membrane are, they have minimal functions, but we talked about them a little bit. And that can bind to the protein receptor that pops out on the endothelial cell. And then what happens is the white blood cell is able to squeeze through those endothelial cells, which, by the way, we contracted and made all that space for them, right? We didn't do it just for that reason, but this is another benefit that's happening here, is now those neutrophils are able to squeeze into the connective tissue space, and the neutrophils also can gobble up pathogens. They also perform phagocytosis. So I actually, because of the size of the capillary, the neutrophils are out of proportion. They're very small. They're not supposed to be that small. They're bigger. They belong kind of more in the same size as the dendritic cells and the macrophages. But that's what they would do. They would gobble up the pathogens. All right. So let's say that what happens next is we go off to the hospital and the nail gets removed, right? And so we still maybe have some residual bleeding. And another thing that, is going to happen pretty quickly, and we're not really focusing on that in this particular video, but obviously we're going to have blood clotting, and obviously we're going to have a scab formation. So we can see our scab, and then we can also see some accumulation of stuff that's right underneath that scab. And so the neutrophils, a lot of times after they engulf pathogens, they're going to die. And so what we get is this accumulation of cell debris, some dead white blood cells, and some fragments of pathogens and stuff like that have been eaten. And that accumulates into this gloppy mess called pus. And I'm sure that you've seen pus before. It's pretty gross, smells pretty bad. And also if it accumulates under the skin, in this case, the scab, it would be pretty easy for that pus to leak out. But if it was a little deeper and a little bit more, we didn't have this injury necessarily on the surface, we'd call it an abscess. In an abscess, you have to actually slice it open and allow the pus to drain out. But the reason that it's so gross and that it smells so bad is because it's actually dead cells that are accumulating there. So we might have that happen. And then soon after that, we'll be able to finally start the process of regeneration. And regeneration, there are two major things that are happening. One is we had this damage to the blood vessel, and we really want to fix it. And so initially, we'll have clotting in the blood vessel, and that will protect against leakage of blood, but we also really want to make sure that the blood vessels are working, so they can try and grow back together, or we can grow some new blood vessels if we need to, but we'll get our blood flow kind of back in order. So we'll have vascular reorganization. And the second thing that's going to happen, and we've also mentioned this cell type before, the fibroblasts, which is another type of cell that's a resident for connective tissue, which the fibroblast's primary job is to produce collagen fibers. And so basically, because we have this gaping wound, we have to kind of fill it back in with stuff. And since we know already that a huge part of connective tissue is collagen, it really makes sense that we have these cells that are just going to make some more collagen, and then boom, we can fix it, and we have this scar formation, or this generation of scar tissue. And I'm sure after having a pretty good sized cut, after looking at the surface of the skin after some time, it's a little pinker than everything else. And then over time we get remodeling, and it will look less and less like a scar and more like regular tissue. And so depending on how big the injury was and stuff like that, affects how much we can see the scar and for how long. But basically, we'll be all put back together, no more nail in the foot, and all this stuff that happened is pretty crazy. But I think that if you really kind of think of it as a step-by-step process, and that all of these things are just really a big synthesis of stuff that we've been working on, I think it will really come together. So I hope you found this helpful, and I hope you enjoyed this very elaborate drawing. And thanks for watching. I'll see you next time.